Welcome to our lesson on chemical reactions and enzymes. Chemical reactions are, of course, not entirely new to you. You spent last year learning chemistry, and we already talked about some important chemical reactions like photosynthesis and cellular respiration. We know that during chemical reactions, substances are changed into different substances, and this happens through the breaking and the formation of chemical bonds. Now, bond energy is the amount of energy that it takes to break a bond. Anytime you break a bond, that requires energy, and anytime you form a bond, energy is released. So here's an equation on the bottom, and this should look very familiar. All right, and just to review some terms involved in chemical reactions, anything that comes on the left side is called a reactant, and anything that comes on the right side is called a product. It's what you're left with at the end. Now, in this particular situation, we're taking oxygen and we're taking sugar and we're breaking those chemical bonds in oxygen and in sugar, and that is going to require energy to be added in. On the right-hand side, you are actually forming bonds. You're forming carbon dioxide and you're forming water, and this is actually going to be releasing energy. Now, this chemical reaction, of course, is cellular respiration, a very important chemical reaction, important for life. Now, reactions come in exothermic and endothermic types. An exothermic reaction releases more energy than it absorbs, and that energy, that extra energy, is often released as heat or light. So exothermic chemical reactions are responsible for the lightning bug's glow. So the fact that the lightning bug glows, this is the product of an exothermic reaction. Pretty cool. The other classic exothermic reaction, and one that we learned about, is cellular respiration, right? More energy is released during cellular respiration than is absorbed, and that's really important because that extra energy is used by cells to do work, right? And also, cellular respiration releases heat, and that's what keeps your body nice and warm. Endothermic reactions are the opposite. They absorb more energy than they release, and an example of an endothermic reaction is photosynthesis, right? And you have the plants absorbing all of this solar energy and using it to form sugar. It's a classic endothermic reaction. The next term I'm introducing you to is activation energy. So as it turns out, just to get a chemical reaction started requires some energy, and the amount of energy you need to get a reaction started is called activation energy. And here's a nice example showing you how much energy is needed to get the reaction started. And it's kind of like pushing a ball up a hill. It takes this much energy to get the reaction started. Once you're on the top of the hill, you can just let the ball roll off, and you can just let the reaction start. This process, or this amount of energy, is called activation energy. Catalysts are things that speed up chemical reactions by lowering the activation energy so that you need less energy, kind of like if you had a smaller hill to push the ball up. Okay, so catalysts are substances that help lower activation energy. Now, catalysts involved in biology are called enzymes. So enzymes are catalysts. They're biological catalysts. We call them enzymes. And the enzymes are needed for almost every single chemical reaction in your body because those reactions would be way too slow to sustain life. You'd be waiting around all day just to get the energy needed for a chemical reaction to start, and life would not be possible. Enzymes help lower the amount of energy you need to get a reaction started. So instead of having to climb this huge hill and absorb this enormous amount of energy, and you can see it with this green arrow, enzymes make it so you need less energy. You have a smaller hill. So you're still starting with reactants, you're still finishing with products, but with the help of an enzyme, you need less activation energy for the same reaction to happen. You can think of this example might be useful. So let's say it's Saturday, it's a really nice day. Let's say it's like a beautiful June day, and there's all these things you could do outside. Like you could go for a run, you could go hang out with friends, but you wake up and you don't feel like doing anything. You just feel like staying in bed and pressing snooze 1,000 times and doing nothing. It takes activation energy to get you out of bed, and that takes a long time. Now, let's say something happens. Let's say your mom starts to make bacon downstairs, or if you don't eat meat, your mom is making cinnamon buns, and the smell hits your nose as you're lying in bed, and all of a sudden, you don't need so much energy to get out of bed, and it's much easier for you to get up, and you're more motivated. Think of the cinnamon bun smell as an enzyme. It lowers your activation energy needed to get you going. Enzymes lower the energy needed to get a reaction started, and they allow them, because you're not waiting around for all that energy to accumulate, they allow the reactions to happen much more quickly. So those are enzymes. Now we're going to talk about how enzymes actually work. So enzymes help in different kinds of reactions. In some reactions, we're trying to build something. So here, for example, we have this molecule and this molecule trying to come together to form this product. Let's say these are simple sugars, right? This could be a glucose and another glucose, and this is maltose, right? And this reaction could be helped by an enzyme. And the enzyme, what it'll do is it'll take these two molecules and hold them close together in the correct orientation so that they can react with each other. Okay, and form this beautiful product. Enzymes also help reactions where you're trying to do the opposite, right? Let's say this is maltose and we want to break it apart into two glucose molecules. In this case, the enzyme will actually use like a little scissor and cut the bond between the two glucose molecules and help that reaction happen. So let's look closer at what enzymes look like and how they catalyze these reactions. So it turns out that the enzyme fits together with its reactants really well. Now, before I use the word reactant again, I want to let you know that we don't use the word reactant when we're talking about enzymes. Instead of using the word reactant, we use this term substrate. So reactants in an enzyme-catalyzed reaction are called substrates. It's what you start with when an enzyme is involved. So an enzyme will actually grab onto a substrate and it fits together with a substrate the way a lock and a key would fit together, or the way two puzzle pieces would fit together. So you can see that the enzyme has a shape that allows it to fit with maltose right here, okay? And this part of the enzyme where the maltose is going to bind, where the substrate is going to bond, this over here is called the active site of the enzyme. All right, so here what's happening is the enzyme is grabbing onto the maltose, and then it's going to cut the bonds between the two glucose molecules and allow the product, which is the two glucose molecules, to now go off and do their thing. The other thing to know about an enzyme-catalyzed reaction is that enzymes are never used up in the reaction. So once the reaction's done, the glucose goes off and does its glucose stuff, and the enzyme is unchanged, and it can actually do another reaction, grab another maltose and make more glucose, and then grab another maltose and make new glucose. Now, there are thousands of types of enzymes inside of your body, and each one is responsible for one chemical reaction, one type of reaction, because the shape is so important that you need a specific enzyme to work with a specific substrate. So here's another example that shows you how enzymes work, and there's a little bit more detail that we're going to cover. So here is our enzyme, this beautiful purple jelly bean, and what it does is it grabs these two substrates here, the orange and the red one, and it's going to hold them on its active site. Now what happens here is this is called an enzyme substrate complex. It involves the enzyme and the substrates, and they're bonded together here. And what you notice happening here is that the enzyme slightly changes shape here to better fit with the two substrates. And then now that the substrates are held together in the proper orientation, the chemical bond forms between the orange and the red molecule. And then the product here, which is made up, you can see the shape looks similar to that shape. The product is released and it's free to go, and the enzyme kind of goes back to its original shape, and it's free to grab another orange and another red molecule and perform the reaction again. And here you see a type of reaction where the enzyme is putting two substrates together, a kind of synthesis type of reaction. Okay, so what are these mysterious enzymes? Because in this chapter, we learned about 4 molecules. We learned about carbohydrates, lipids, proteins, and nucleic acids. It turns out that enzymes are an example of a protein. And you might have already remembered this from our earlier lesson when we talked about the jobs of proteins. And here are all these protein jobs, like they make muscles, hormones like insulin, they fight disease during flu season. And then the other thing that proteins do is they act as enzymes, right? So one function of proteins is to be enzymes. So enzymes are proteins. That means something though. Remember, proteins change their shape when they're at the wrong temperature or the wrong pH, and their shape is super important for them to function, right? And we saw that enzymes, their shape absolutely matters because they fit with their substrates like a lock fits with a key. So if they're at the wrong temperature or the wrong pH, just like proteins, enzymes will warp and they will no longer work, right? Remember, the proteins changing shape in the egg causes them to, to go from an egg that's runny to a hard-boiled egg. That's the denaturing of proteins. All right, and it happens also when we're forming basically curdled milk. All right, so I'm going to show you what happens when the temperature and the pH of an enzyme catalyzed reaction changes in this little video. All right, so here what we have is our enzyme on the bottom in green. And we're going to start to see, here is our substrate flying in. And you see it doesn't fit too well with the enzyme. And here we're changing the pH or the temperature. And as that pH or temperature is going up, we're getting closer to the right temperature and the right pH. And the enzyme is changing shape to fit better with the substrates. And look at how fast the reaction is going now. It's going really fast because we're at the right temperature or pH. And then the temperature and the pH starts to get too high, and the reaction slows back down again, and we start to slow down. So the thing to know about this is enzymes are proteins, and proteins have a specific temperature at pH at which they work the best. And for most enzymes in our bodies, the pH that they work best at is a pH of 7, which is the pH that we have in our bodies. So I really hope that was helpful, and it was a good introduction to enzymes. Thank you so much for watching.